Hi guys. In this video we're going to continue our discussion of signal transduction in the nervous system. In the last one we talked about how an action potential might propagate along an axon or dendron, but eventually you come to a synapse, i.e. a gap in between different neurons, and in today's video we're going to talk about how the signal crosses that gap. That gap is called a synapse, so we'll be talking about synaptic transmission today, okay? Synaptic transmission. Now, just a few reminders and a few new structural things to talk about before we begin. First of all, don't forget that in this extracellular space we can't ignore still, even though we might not talk about it too much today, we can't ignore the continuing contribution of the sodium-potassium pump. Okay, so that resting potential is not just along axons. You've got to think of that resting potential as existing, along all the membranes of all the neurons. Okay, so in this extracellular space, we've got lots of sodium. Within all the cells, we should have lots of potassium. Okay, and we can't forget that that's because of the sodium-potassium pump. All right, so I'll just put that here. Sodium-potassium pump. Right. Okay. Right. So we're happy with the idea that these are voltage-gated sodium channels. Okay, voltage-gated sodium channels. These will represent voltage-gated potassium channels. Okay, and these are representing today, and these shapes have no particular significance. I don't know what they really, what their shapes are really like. I've just drawn them different so that we can discriminate between them. Okay, so these are voltage-gated calcium channels, right? Calcium. So synaptic transmission involves the addition of the new ion, which is calcium channels, but we'll see how that works. Okay. Right. Okay. So just a few things with the synapse, because that's really the new structure that we're going to be talking about. This area here is called the. So this area right here is called the synaptic knob. This is called the synaptic cleft, so the kind of the space, the gap between the two neurons is called the synaptic cleft. Okay? We'll refer to this membrane as the presynaptic membrane. We'll refer to this membrane as the postsynaptic membrane. Okay? And this neuron will be the presynaptic neuron. This neuron will be the postsynaptic neuron. Okay? These gates are ligand-gated sodium channels. So they are sodium channels. They are sodium channels, but they are ligand-gated and not voltage-gated. Not voltage-gated like the ones along the axon or dendron membrane. Okay, these are ligand-gated, and we'll talk about what that means and how they behave differently because of that. Okay, right. Okay, these are vesicles, and within the vesicles we have a neurotransmitter. But okay, so we have all our characters here. We'll talk about what they do in a second. So let's begin then. Remember, we have lots of sodium out here in the extracellular space. Okay, and that's all thanks to the sodium-potassium pump. Okay, I'm not going to do it down here, but it's the same thing happening at the bottom. Right? Lots of sodium. I'm sure you get the idea. Okay. Right, and we cannot forget that we have lots of potassium inside. Okay. And we also happen to have calcium ions. I'm not putting the charges on, just to save on time, but sodium ions have a +1 charge, potassium ions have a +1 charge, calcium ions have a +2 charge. Okay? Right. And now we can begin. So, action potential arrives. We have a depolarization here. And this is going to be a bit of a recap for you. But the depolarization causes voltage-gated sodium channels to open. Sodium comes in. Sodium comes in. Eventually, when you reach the action potential in this region, the potassium will go out. Okay, so be localized. Okay, you'll have lots of potassium out there. It'll be gone from here. Okay, and that will be the repolarization. Okay, and that will cause the next set of voltage-gated sodium channels to open. So sodium will come in here. Potassium during the repolarization will go out. Okay, as you know, following that, so we'll have potassium out here. We'll have lots of sodium in here. But remember, here the repolarization will have proceeded even further, and both gates will now be closed, and the sodium-potassium pump will now be working to re-establish that balance. So behind this depolarization, everything behind is getting back to normal. Okay, all the sodium is being pumped back out, and the sodium transported, the potassium transported actively in. Okay, so behind the depolarization, so once you've started the depolarization in the next step, behind recovers. Then the depolarization spreads over here, the action potential spreads over here, okay, and eventually even this part will recover, all right? But here's the problem. You can see that if we carry this sequence along, eventually we're going to run out of membrane. We're going to run out of membrane, we're going to run out of cytoplasm. Essentially, it's the end of the cell. We've got a gap that we can't cross with an action potential, okay? And this is where synaptic transmission comes in, because it uses a slightly different mechanism to get the message to the next cell, and then the next cell will then carry on the depolarization wave, action potential wave. Is that making sense? Okay, so what happens next? So we had a depolarization here. It caused the depolarization in the next part of the membrane, while this part is now repolarizing. Now we've got an action potential here. What happens next? Well, the depolarization here opens voltage-gated calcium channels. Okay? So the depolarization causes voltage-gated calcium channels to open. This means that calcium comes in to the cell, diffuses into the cell. Okay? No doubt there's a concentration gradient, and the calcium was just waiting for the channels to open and move down its concentration gradient. So the calcium comes into the cell, and remember, the calcium is only there because these channels opened, and those channels only opened because there was a depolarization spreading along the neuron. Okay? So when that depolarization got to the voltage-gated calcium channels, they opened, calcium moved in. Now when calcium ions come into the cell, they start a series of signaling reactions that causes these vesicles to move towards the membrane. Now, these vesicles, they're going to move towards the membrane. All right, so maybe I'll draw one of them getting very close to the membrane, like so. And within those membranes is a neurotransmitter, okay? And that neurotransmitter is called, you see that? Acetylcholine. Okay? I'll get rid of the other information I don't need. Okay, so the neurotransmitter is called acetylcholine. Very small molecule, okay, but it's lots of it packed in these vesicles, just waiting to be released into the synaptic cleft. So they're just waiting there, these vesicles, but when calcium enters the cell, because of the change in voltage caused by the action potential arriving, those vesicles start to move towards the membrane, and eventually they fuse with the membrane. So the membrane of the vesicle joins with the vesicle of the membrane of the presynaptic membrane. So the vesicle membrane fuses with the presynaptic membrane, and when that happens, when those membranes fuse together, right, they become essentially one membrane that continues all the way around like this, meaning that the acetylcholine is released into the synaptic cleft. That's very important. So the acetylcholine is then released into the synaptic cleft, it diffuses into the space, and because of this diffusion across this gap, it kind of slows the signal transduction down a little bit, okay? So the action potential is moving quite fast because of saltatory conduction, because of the myelin sheath, jumping from node of RON-VA to node of RON-VA, jumping, okay, calcium comes in, vesicles released, but then we've got to wait a little bit for the acetylcholine to diffuse across the space, and that slows the transmission down a little bit. So now we've got our acetylcholine diffusing across the gap, and eventually it's going to bind the ligand-gated sodium channels. Okay, ligand-gated sodium channels. Ligand is something that binds to a receptor. Okay, and in this case, the ligand is our neurotransmitter acetylcholine, and it's going to bind to the ligand-gated sodium channels. Now remember, voltage-gated sodium channels opened and closed in response to changes in voltage. Ligand-gated sodium channels open and close in response to whether or not the ligand is bound or not. Okay, so our acetylcholine diffused across the gap, bound the ligand-gated sodium channels on the postsynaptic membrane, and when the ligand binds there, when the acetylcholine binds those ligand-gated sodium channels, those sodium channels open. Okay? The sodium channel is now open. And remember, we always have a high concentration of sodium outside the cells. Okay? And it's true just in the synaptic cleft, and it's true there just as it is for the axon or dendral membrane. Okay? So when the acetylcholine binds that ligand-gated sodium channel, it opens, sodium then rushes in comes sodium, the sodium concentration in this area increases, and if it increases enough, it will cause a depolarization in the postsynaptic neuron. And if that depolarization is large enough, it might not be, if that depolarization is large enough, it will cause these voltage-gated sodium channels the same as these, but here at the start of the next neuron, it will cause these voltage-gated sodium channels to open and cause even more of an influx of sodium into the cell. Okay, now what we've got to think of is our graph again, and I hope you can see this. We've got to think of our graph again here. what we said was, if you reach the threshold potential, then you'll activate an action potential. Okay? So, if our resting potential's here, a little bit of sodium coming in might cause a depolarization. But we need loads of sodium to come in. Okay? So if lots of sodium comes in, then we'll get an action potential. Okay? Because then we'll have reached the threshold potential. Alright? And then it'll come back down. Stabilize. Okay? So the idea here, we can just recap again, is that the depolarization spreads across the neuron by saltatory conduction, but when it gets to the end, it can't jump across the gap. It needs cytoplasm, it needs membrane depolarization, and the membrane here stops. Okay? And we've got a gap. So the gap, the signal passes on by diffusion of a neurotransmitter across the gap. Okay? Right, so when the action potential spreads up to here, calcium, voltage-gated calcium channels open, calcium enters the synaptic knob and causes the vesicles to move towards the presynaptic membrane. Those vesicles fuse with the presynaptic membrane, releasing their neurotransmitter into the gap, into the synaptic cleft. That neurotransmitter, in this case acetylcholine, diffuses across the gap, binds the ligand-gated sodium channels, they open, they let sodium come in, move in, diffuse along their concentration gradient, and if enough sodium comes in and causes enough of a depolarization in the post-synaptic neuron, voltage-gated sodium channels will open here, cause more sodium to come in, and if enough sodium comes in and if the depolarization reaches the threshold potential, we will get the initiation of an action potential in the postsynaptic neuron, which will then move all the way down once it's been started. Okay? And that's all I have to say about that. Yeah. Okay, so we've discussed how an action potential moving along the axon or dendron, well, this would have to be the axon, moving along the axon will then eventually reach the end of the cell, okay, the presynaptic membrane, and the action potential can't go along, so it has to move across using the neurotransmitter to diffuse across the gap, bind to the ligand-gated sodium channels, cause them to open and allow the depolarization to start in the postsynaptic neuron. But what we need to talk about next is what happens to the acetylcholine. It can't just remain bound to the channel, because it's going to let lots and lots of sodium enter the cell, and we don't want to keep firing a signal when, you know, all we needed actually to do was to, you know, make a signal in response to the signal that was coming down here. So we need a way to switch off the signal and prevent overstimulation of this postsynaptic neuron. And the way we do that, or the way it happens, is by the presence of an enzyme that breaks down the acetylcholine in the synaptic cleft. Okay? Now that enzyme is called, rather conveniently, acetylcholine esterase. Okay? Acetylcholine esterase. And what it does is it cleaves the acetylcholine that's bound to the receptors, breaks it in two, and it's then no longer in the right shape to be able to bind it's then no longer in the shape that's needed to bind the receptor. Okay, so it kind of broken. All right, and because of that, the sodium gates will then close. When they close, then there's no more depolarization occurring in this area, and by which time, you know, the actual action potential that was initiated is way down the neuron. Okay? Now, what happens next is that, we don't waste this acetylcholine, or at least we don't waste the choline part of it. There's a special mechanism, there's a special route where the choline part of the acetylcholine can re-enter the cell, become reformed into acetylcholine, and become packaged in new vesicles. Okay? But remember, this process requires ATP, which ultimately comes from the mitochondria. And that's why you'll see lots of mitochondria in this synaptic knob, because it's needed to reform the acetylcholine so that when another signal comes along, if another signal comes along, it's ready, the vesicles are ready to release their contents again into the synaptic cleft. Okay, so that's one thing that's very important. Next we'll talk about the signal, the strength of the signal needed to start an action potential in the postsynaptic neuron. Okay? It might not always be the case that one action potential comes along, acetylcholine released into the synaptic cleft, action potential started in the postsynaptic neuron. It might not be that straightforward. The system might be set up so that one action potential is not enough. Okay? So the release of some acetylcholine into the space, into the synaptic cleft, is not enough. So say if this vesicle releases its contents, enough to open one sodium channel, you get a little bit of sodium coming in. Okay? But that little bit of sodium is not enough. It depolarizes the postsynaptic neuron a little bit, i.e. the potential inside does become a little more positive, but it doesn't reach the threshold. That red dotted line there, it doesn't reach that threshold. Okay? So it might be that we need repeated stimulation, so another action potential to force another vesicle to fuse, or more vesicles to fuse with the membrane, more acetylcholine to be released, open more channels, and let more sodium come in, and then we get a little bit higher depolarization, and then only, it's only then that we reach the threshold potential, and that causes the action potential, and then depolarization, and then stabilization, hyperpolarization, and polarization again. Does that make sense? Can't hear you. Yeah. Okay. So it might be the case that you need repeated signals to release enough acetylcholine to then open enough voltage-gated sodium channels to then let enough sodium come into the postsynaptic neuron to then reach the threshold potential and then cause the action potential. Okay? Well, now that, when you need multiple action potentials in order to do this, this is called a type of summation. It's called temporal summation. Okay? And I guess it's a way to make sure that it's real stimuli that are it's information about real stimuli that's causing messages to be transmitted across the body, not just random firings of action potentials. Okay, it has to be a real stimulus, and a real stimulus will cause repeated action potentials. Okay? That's that. There are some aspects that I'm not going to go over, like the roles of synapses, but I think this is the key concept. If you get this, then it will make the other things fall into place. Alright guys? Good luck. Again, any questions, leave a message and I'll respond as soon as I can.